Dear students, Assalamu alaikum. Today we are going to talk about our course Special Topics in Biochemistry, course code CHEM 6462. This is lecture number 3. This lecture is part of our series of lectures on the topic Contemporary Biochemistry and Molecular Biology Techniques. The outline of today's lecture is we will talk about what is immunohistochemistry, abbreviated as IHC, and we will talk about the steps required for conducting immunohistochemistry experiments. We will talk about general structure of antibodies. Understanding the general structure of antibodies is important in the sense because immunohistochemistry and immunocytochemistry, they require antibodies, and understanding the structure becomes very important for understanding the process of immunohistochemistry and immunocytochemistry. So we will briefly talk about how the general structure of antibodies is and how their specificity, the powerful specificity properties are helpful in conducting these experiments. We will talk about direct and indirect methods of immunohistochemistry. Sometimes people use direct, while in other cases, in majority cases, they use indirect methods for immunohistochemistry. Then in the finally we will talk about immunocytochemistry. Immunocytochemistry is basically similar to immunohistochemistry, but in immunohistochemistry generally we look for tissues, while in immunocytochemistry we study different proteins in a cell culture environment where cells are used, and so this is the difference as well between the two techniques. Otherwise, there are a lot of similarities between the two. This is why we are studying these under a single heading in a single lecture. So let's go for the topic. Now, the first question that comes to mind is, what is immunohistochemistry? Immunohistochemistry is the localization of a known antigen in tissues by utilizing antibodies directed towards that specific antigen. Now, I would like to remind you about PCR that we studied. We talked about PCR in detail and how the different PCR reactions tells us about a gene product. How PCR reaction tells us about amplification of a certain segment of DNA. Now, according to the central dogma of molecular biology, we get from DNA, we get RNA, and then we get proteins. So in case we study certain gene whose DNA is produced, now we want to know if it has translated to a protein or not. Immunohistochemistry is the technique of choice. It will tell us about if the protein has been has been formed or not, and then with the help of immunohistochemistry, we can also localize the protein that in which part of the tissue the protein is localized. So now in this lecture, in this slide, we are going to talk about the steps that are taken for conducting immunohistochemistry experiments. First of all, for conducting immunohistochemistry experiments, we will need to prepare tissue sections. The tissue sections are prepared with the help of microtome, or with the help of in case of in another case, if we go for cryosectioning, we also have we can also get tissue sections. So preparing tissue sections is the first step for conducting immunohistochemistry experiment. Once we have the tissue sections prepared, the sections are processed for antigen retrieval, and after following the protocol of antigen retrieval, what we do is that we block the endogenous enzymes by using certain material like BSA. With the help of this, the endogenous enzymes, they are blocked. Then in normal situations, we use primary antibodies. The primary antibodies are the antibodies that are directed towards the antigen or the protein that we are looking for. Then after this, once the primary antibodies gets attached, that the sections are thoroughly washed. Now you have to understand this, that once primary antibodies are attached with its antigen, even if we wash the tissue sections, the primary antibodies, they don't detach from the antigen because their binding is very efficient and very and they cannot be washed away. And in case the primary antibody, and in case the antigen is not present in the selected tissue sections, the primary antibodies will be washed away, and so we will not be able to detect anything. So this antibodies specificity is very important in conducting immunohistochemistry experiments. The other step is secondary antibodies. Using secondary antibodies, secondary antibodies are the ones which are specific for binding of primary antibodies, which means that secondary antibodies can only attach with the primary antibodies. Now, in case we do immunohistochemistry experiment, and the primary antibodies are not attached because the antigen against which they were directed was not present, secondary antibodies will also not be able to attach because the primary antibodies will be washed away. Now, in case the antigen that we are looking for was present, primary antibodies will react with that, and they will strongly bind with the antigen, and even if we wash many times, the primary antibodies will not be washed away. and when we apply secondary antibodies, they will be able to attach with the primary antibody. These secondary antibodies, they are generally attached with a chromogen substrate or with fluorescent material, and when we look at the sections through fluorescence microscope, for instance, we will be able to visualize them. The visualization of the secondary antibody will tell us that primary antibodies are present, and the primary antibodies presence shows that the antigen or the protein that we are looking for is also attached. So basically, when under fluorescence microscope, when we look and we find fluorescence, we see the fluorescent material that is attached with the secondary antibodies, and thus we can say that primary antibody has reacted with the protein and the protein is present. And then we counterstand, slides are mounted, and this is after doing all this protocol. The sections of the slides are observed with the help of microscope. So this tissue summarizes the overall process of immunohistochemistry, and stepwise it is performed as in a stepwise manner. And this is how the assay looks like. This is in case of cells, for instance. Cells are grown, they are spun into a pellet, they are either frozen or paraffin embedded, and they are sectioned. And sometimes the cells can be grown as a monolayer, like this, a single layer of the cells. Now you have to understand that why we need to make thin sections, because they are helpful in many ways. One, through sectioning we are able to visualize the cells under microscope. Another one of other reason is it is helpful because the antibodies they can reach the cell easily, because you know that normally cells are covered by the membrane. So if we don't prepare sections, it will be difficult for antibodies to reach inside the cell. These cell membranes are ruptured with the help of different types of detergent-like material, like Twin 20, for instance. So they are helpful to provide a passage or a path for antibodies to reach inside the cell. And once antibodies they reach the inside the cell, they are able to attach or react with the antigen that we are looking for. And you have to understand another point here is that the immunohistochemistry technique is very helpful in the sense because the immense power of antibodies to be able to specifically bind to their antigen is very helpful in this technique, because in antibodies that are produced, they only specifically bind bind to the antigen against which they are produced. So there are very few chances of unspecific binding. In other cases, in cases of tissue, we use tissue sections that are either frozen or paraffin embedded. Now, you know that in sections from tissues, they contain many different kinds of cells as well as extracellular matrix. You can see here, this is a tissue section, and we expect from tissues that they will contain different kinds of cells, and they will also have extracellular matrix in which the environment in which the cells are there. So after sectioning the tissue, the cells are embedded in the tissues, and there are, you can see that there are different types of cells, and we also can see that the different extracellular matrix components. So this is how the cells can either be like this. This is what we are talking about, cells on the slides, or tissue sections that are frozen or paraffin embedded. So cells can either be like this. This is in case of cells, and this is in case of tissues, where different types of cells and extracellular matrix is also present. Now, before going further, we have to understand the general antibody structure. The basic structure of an antibody is it consists of light chain, the one that is in green, and it consists of heavy chain, the one that is in red. So you know that the structure of antibody is such that it consists of two chains, one the red heavy chain and the other light, the other green, it is the light chain. And these two chains, they are also linked with each other through the disulfide bond. So there are different disulfide bonds that are present, and thus this is an antibody connected through disulfide bond consisting of light chain in the green color and heavy chain in the red color. Along with this, in antibodies, we have two regions. One, this blue color is constant region. So this part of the antibody is called constant region, and there is this red part. This is called the variable region. So from antibodies to antibodies, among different antibodies, this region can be different. So antibodies, to summarize the whole story, antibodies consist of light chain and heavy chains, and then they consist of constant region is shown in the blue, and variable region is shown in the red. So this is important to understand the process of immunohistochemistry. So hopefully this through this slide you are able to understand the general structure of an antibody. Now there are, as I told in the outline, that there are two ways that antibodies, that immunohistochemistry can be conducted. One is the direct method, In direct method, this is the antibody. In the direct method, we don't use secondary antibodies. The fluorescent material or the chromogen is attached directly with the primary antibody, and once the primary antibody, it attaches with the antigen, and once we complete the whole protocol of immunohistochemistry, one can visualize the antigen by this method, and we are able to see if the antigen is present or not. But in majority of cases, indirect method is used. In indirect method, we use both primary and secondary antibodies. So this is the antigen. What happens in indirect method is that the primary antibody, it comes and it attaches or it reacts with the antigen, and after it reacts with the antigen, we apply secondary antibody, and the secondary antibody, they are specific to the primary antibody. It means that it attaches to the primary antibody, like as I told you, that when we conduct the immunohistochemistry experiment, we wash these sections multiple times, and if the antigen is not present, the primary antibody will be washed away. But when the antigen is present, the primary antibody reacts with the antigen, and even if we wash the sections several times, the primary antibody will not be washed away. And when we apply secondary antibody, the secondary antibodies are specific to primary antibody. It means that the secondary antibodies are able to attach with the primary antibody, and the secondary antibodies, they have this fluorescent material attached to it, and using secondary antibodies is useful in another way, that it helps in increasing the signal intensity. What is meant by this signal intensity? It means that the signal that we will get is enhanced, because the primary antibody has the ability to attach or to bind with multiple secondary antibodies, and this enhances the signal that we will get, or we will receive on the we will get on the microscope. So you can see that the fluorescent material, this is because through microscope we can't directly see the antigen, we see the fluorescent material. So these fluorescent materials, we will be able to see them more clearly if we have more intensity coming out of here. So this is a way to get a good clear resolution, a clear picture of the fluorescent material. So this these two slides, they summarize the overall process of how immunohistochemistry can be conducted. It can be done either through direct means, like I showed you here, in which the material was attached directly to the primary antibody, and in this case where the fluorescent material is attached with the secondary antibody, and we get better fluorescence. Now this is a practical example of how it works. This is a real-time PCR data. It shows that transcription factor ZNF142, it shows a significantly higher fold change as a result of real-time PCR. We see that it's as compared to the control, it shows a very big fold change. And when we do these are the results of the immunohistochemistry. When we do the immunohistochemistry for the control and for the 3 days after castration, in this situation, this is published data from one of our article in a very prestigious journal of cellular physiology, published in 2019. The result that it gives that it shows that this is the negative control. We see that there is no ZNF. Sorry for this. This is ZNF142. So we can't see anything in green. So it shows that in the control we can't see any ZNF142, while after 3 days, if we do the immunohistochemistry, we can clearly see these cells expressing ZNF142, and ZNF142 can be seen in multiple cells very clearly. So it shows that the expression of the gene has translated into protein. It means that from DNA is not only messenger RNA has been formed, but it has also translated successfully into a protein, and one can see that it is mainly localized in the epithelial cells of the prostate in this case. So the details of these experiments can be seen in the paper if you study this in greater detail. But this gives us an idea of the expression that not only messenger RNA of the protein has been produced, but it has also translated successfully into protein, showing that the protein has been produced. And this is the power of immunohistochemistry that is giving us these information. Now this is in another work this is another experiment which was done for immunohistochemistry, and this immunohistochemistry is giving us information about neonatal prostate development. This is another transcription factor that is called ZNF703, and in this situation the antibody was diluted 200 times. Now we can see that in the negative control we only see the DAPI, the standing for the nucleus. The blue color that you are seeing is the nucleus of the different cells, And when we see after 6 days, for instance, we clearly see this red. In this situation, the color of the fluorescent material that is used is red. So this is why we see this red region. It shows that all these cells which are showing the red, they are expressing this protein ZNF703. So this is how, in another picture you can also see that ZNF703 is being expressed, and these pictures were taken through a confocal microscope, and this work was also conducted by us and published in prestigious journal called Life Sciences in 2015, and you can read this complete paper. Then this is the DOI of that paper. So also, here also it shows that immunohistochemistry has the power to localize, not only to be able to see the expression of protein, and also localize the cells which are expressing certain protein. Because through PCR or through real-time PCR, we can only get information about how if the gene is expressing. But even if a gene is showing a 4 gene increase, we can only confirm by the help of immunohistochemistry if the expression of the DNA is really translating into a protein. So this is a very helpful technique. Here is another example that I would like to share with you. This is a transcription factor called DMRT2. This DMRT2, this is the real-time PCR result of DMRT2, and this is the control. This is day 1 after castration, day 2, 3, 4, 5, and 5. One can see that it is showing statistically significant expression. And when we conducted the immunohistochemistry experiment for day 5, we can clearly see that this DMRT2 is expressed. In this situation, the fluorescent tag material is green in color, and we can clearly see through the arrows that protein is not only showing a 4 gene increase, but it is also translated. This work has already been published in another journal called Histology and Histopathology in 2019, and you can read this complete paper from internet. You can search it and you will be able to find. If you want me, I can share with you the complete paper. So it also shows that it is indicating, or it is showing that the real-time PCR results, that the gene expression is not only showing increase, but the translation of the gene is also taking place, and it can be localized in the tissue section as well. Now finally, I would like to tell you about immunocytochemistry. This is an example of RWPE1 cells. for ZNF142 transcription factor. The cells were grown in the RWP1 cells were grown. This is the blue color is showing the Doppy staining, the staining which stain nucleus. So there is nothing green here because this is the negative control here, and these cells when they were stained against antibodies ZNF142, we can clearly see that these cells, they are expressing ZNF703, the color of ZNF703, and here is this. So this is also how immunocytochemistry are. When we conduct this staining with in cells, we are able to see how it can successfully help us to visualize if a certain protein is being expressed or not. I hope you enjoyed the lecture, and if you have any questions, I will be very happy to answer it. Thank you very much for your attention. See you in the next lecture.